My name's Jo Cambridge, and I'm an immunologist at UCL, where I work in the rheumatology department. I'm going to introduce Fane Mensah later, who's an IIME-funded PhD student in his first year of his PhD, and we work with two clinicians at two different centers. I'm not going to give you data today. I want to tell you about Rituximab. Okay, so Rituximab has been used in the last 15 years as an immunotherapeutic agent in diseases other than malignancy. It was designed to be used in lymphoma, but Professor Edwards started using it in rheumatoid arthritis in the late 1990s. Okay, so I do everything Rituximab in rheumatoid arthritis, lupus, some hematological diseases, and we look at all aspects of B-cell function and other molecules as well. So I'm pretty Rituximab'd. Okay, so I'm a relative newcomer to ME/CFS, but I came into the field accepting fully that ME/CFS was an organic illness, and from the Norway study and from other studies in the past, the suspicion is that products of the immune system are involved. Okay, this is a really oversimplified version of what we think, the model we work on. The brain and the immune system share so many receptors, so they're intimately involved, of course. There are obvious ways that antibodies to brain or whatever can influence, can cause disease, but it's complicated, as you saw. And Carmen's described antibodies to receptors, and these receptors are also on, these beta-adrenergic receptors, as she said, are also on B cells as well as on other parts of the body. Okay, so in ME/CFS, the Norwegians have now done 3 studies, and it does seem to work in some patients, and we work with Flugenmella-2 in Norway. So what could B cells be doing? Getting rid of B cells seems to work, so what could be the role? It could be that the antibodies produced by B cells, as Carmen suggested, are interacting with neuroendocrine antigens or other mediators in the nervous system. But also, it could be through inducing cytokine production with not just T cells, which they usually interact with, but other tissues as well. So I'm just going to very quickly go through some of what we've learned in the last more than 10 years. I'm trying to condense it down. What does the treatment consist of? How soon does it work? How do we monitor disease and other disease response in other diseases? Why does it work in all patients? And how patients relapse, because in autoimmunity, many patients do relapse depending on the disease. Okay, so I'm sure you all know that Rituximab binds to and kills CD20 positive B cells. CD20 positive, it's a monoclonal antibody, which is mostly humanized. It's got a little bit of a mouse in it, and it binds to the CD20 molecule shown here at a particular site. Obviously, it's a monoclonal antibody. There are other anti-CD20 agents that bind to different sites and have slightly different abilities to kill the B cell. There's many of these now. So I'm just going to go through a very brief version of the life history of B cells so you understand where the Rituximab is working. So B cells are made continuously by the bone marrow. They're coming out of your bone marrow all the time. And then they migrate through all the tissues, particularly the lymphoid organs, and look around for bugs, viruses, whatever, for signs of inflammation. And once they find an antigen in the context of other signals, they interact with T cells, and the B cells can then undergo clonal expansion and formation, firstly, of usually low affinity antibodies from plasma cells. And then if they get good help from T cells, they can form germinal centers and make antibodies that can actually be produced by these long-lived plasma cells for many years, up to 17 years. Okay, so which cells are depleted by Rituximab? Virtually all the peripheral B cell compartment, in pink here, are CD20 positive, and these are all killed. But these early ones that are coming out of your bone marrow, just about to come out of the bone marrow, these don't have, these have got a little bit of CD20, but these don't have any. So you're not getting rid of your sort of stock, as it were. And plasma cells don't express CD20. Okay, so these earliest B cells and plasma cells are not depleted. So this can be a problem when your antibodies you want to get rid of are made by these cells, but maybe we could talk about that. Okay, so what does it consist of? It's not a pill. It's an intravenous infusion of doses, usually 1 gram, a week or 2 apart. And then the patient goes away, as long as they haven't reacted in any way. Infusion reactions are rare, but some patients do get a little bit of a few reactions, and then you wait. And we have a blood test a month to see if the B cells have been removed successfully. So there are 2 main mechanisms that rituximab uses to kill B cells. This is the rituximab here. This here represents a thing called complement, which lyses B cells. But mainly, rituximab recruits phagocytes, and these kill the coated B cell. So, sorry, that was my son. Sorry, he had to put that in. Okay, so what I've shown here is how we actually look at B cells. Now, every dot here is a B cell, and they've been stained with 2 different antibodies called, you don't have to worry about this, IgG and CD20. And it allows you to divide them into naive, they're the early ones coming out of the bone marrow, and the memory population. These ones here are all memory, and these ones up here are naive. And you can see after rituximab, this is about, I think this is about a month after rituximab, you can see that they've all been depleted, or most of them. You often find remnant memory cells. Memory cells are a little bit more resistant to rituximab, but it's complicated. So, rituximab kills B cells very quickly, within days, but it kills tissue B cells at very different rates in different tissues and in different patients. And in fact, a lot of B cells are left behind, but it doesn't seem to matter for most diseases. And it particularly persists for a long time in the bone marrow. And so when it's there, it stops all the new B cells coming out. The new B cells in most patients come back around 6 months after rituximab. So they start coming back out again. Poor clinical response in patients with autoimmunity is often related to the B cells not being killed properly by the rituximab. And we've shown in lupus, for example, the lupus B cells take in the rituximab and destroy it inside the cell. So it's a very disease-dependent thing, but there are newer reagents that don't suffer from this problem. So rituximab, when you're looking for predictive factors for whether a group of diseases is going to work, the best has been shown to be the presence of autoantibodies, which we proposed many years ago, and where autoantibody was obviously a part of the disease process. And so all the ones in red here, these diseases, there are no autoantibodies associated with these diseases, and people have used rituximab, and it doesn't seem to work. Most of the other diseases where there are autoantibodies, it's been tried and with great success in most. So rituximab, therefore, particularly with autoantibodies as part of the disease process, it functions to remove the parents of autoantibody-producing plasma cells, for example. So, and usually, fortuitously, most of these plasma cells are actually rapidly turning over, so the B cells are differentiating really quickly, so they're more likely to make an effect on the autoantibodies. And also in the case of autoantibodies that form immune complexes, for example, in rheumatoid and lupus, it stops the supply of the autoantibodies making these immune complexes, which are pro-inflammatory. Okay, so the success that we've seen in the Norway study may therefore, if you look at backwards, imply perhaps that autoantibodies are part of the disease process, as Carmen has suggested. Okay, what about the timing of response? People were worried about this because they think often that the response will take place immediately when you get rid of the B cells, but that's not the case. It can take months. It virtually always takes months. And if you think of the immunoglobulin's half-life in blood is about 20, IgG is about 28 days for some of the subclasses, but it will also, you've got to knock down the antibody supply to a manageable, non-pathogenic level. So, and it also depends how the autoantibodies are actually interacting with the antigen in the tissues as well. So you can imagine it can take a long time, or months. Okay, and so people say, oh, all the patients are going to die of infections if you give them rituximab. This has not turned out to be the case. Again, fortuitously, antibodies in the blood to pathogens respond in different ways in quite a lot of the cases than autoantibodies. It depends on their origin. For example, as I've said, the ones that are turning over quickly will be got rid of, but the ones from long-lived plasma cells, not so. So now I've told you about how it's very variable, the time that people respond. So you can't sort of look at responses at 1 month, 6 months, whatever, because people respond differently. So we use a different sort of criteria for sampling. We use baseline, when the B cells are depleted, when the patients repopulate with B cells and maybe relapse, or a time after when they relapse. I just want to show you a little tiny bit of data. This is the different effect of rituximab on antibody levels in rheumatoid patients. If you look at the top one, so this is pre-treatment, when they're depleted, when the B cells come back, and when they relapse. And you can see these autoantibodies go down and then come up at relapse. But these protective antibodies, and these are against pneumococcus, this is 3 cycles, over 3 cycles of rituximab. It's a patient followed up for 10 years. And the protective antibodies really do not go down significantly. They're preserved. And that's because these, like TET-tox antibodies and these ones, are made from long-lived plasma cells in the spleen and bone marrow, whereas these are being generated all the time from new B cells coming out of the bone marrow. Okay, so when rituximab, so what determines B cell return? Well, we think it's because it relates to when rituximab is cleared from the bone marrow. So once it's cleared, then the new B cells coming out and developing CD20 on the surface stop being killed. And when they come back, it's just like being born again as a B cell. So again, this is one of the FACS plots. This is slightly different. These are naive cells up here, and these are memory cells down here in this one. Now this is before rituximab, and this is when the B cells are coming back. And you can see they're repopulating by this population here. These are naive transitional cells. These are just out of the bone marrow, and this is down to 7% mature post-germinal center cells. And this is more than 4 months after the B cells have come back, and they're moving this way, so they're going from transitional to mature naive. These are still not coming up very much. They're accumulating here. So this is what you see when the B cells come back. And relapse is not always associated with B cell return. This is something that was perhaps a bit of a surprise early on, but not really. Just looking at this red line here, this is a measure of inflammation, so disease activity. Disease activity has gone down, the patient's still well. These are the B cells coming back here, and the patient is still well. So in rheumatoid, about 70% of the patients respond. It takes them more than 4 weeks to respond usually. The B cells come back around 6 months. And about 65% do relapse when the B cells come back, but others have much longer reductions in disease activity. So are there any sorts of B cells that trigger relapse? Well, I could go on about that, but I'll just say that some B cells trigger relapse when the new B cells engage certain pathways. In other patients, the B cells need to mature and actually make different antibodies before they relapse. Right, rituximab and ME/CFS. Why it may work? Well, we've seen that Carmen's shown that it looks like the B cells, the rituximab is stopping the B cells differentiating to plasma cells because you saw that reduction in the antibodies. And it could also be in something where it's in the way the B cells interact with other cells, making cytokines that actually stimulate the production of autoantibodies. The other thing I'll just touch on is that it's very important when you're investigating a disease and the effect of rituximab is where the autoimmunity is hardwired, whether it's hardwired in these new B cells coming out of the bone marrow, or that it requires T cell help and it's dependent on interactions within germinal centers. So, as I suggest by that last slide, if you know what's going on, you can adjust your treatment protocol depending on how the B cells are involved in causing the disease. So it's disease specific. So at UCL, we've been looking at both naive and memory B cells and the effect of T cells on these, and with the hope of finding out where things are going wrong in ME/CFS. So our approach is to look at, in detail, at the phenotypic markers on the B cells, and also look at metabolic changes related to these differences, and then also, most important, to model possible interventions to the immune system in ME/CF patients' cells in culture. So Fane Mensah, our PhD student, is going to take you through some of the work we've been doing at UCL. Thank you.
Thank you for the introduction, Joe, and it's really pleased as a PhD student to present a little bit of my work I've been doing the last few months. An important thing that I want to address is that we do a lot of research, but we also collect clinical data on the patients, because some of the patients who've been here know that we collect also some questionnaires on the symptoms, which we eventually later on also want to correlate with the things we find in vitro. So this is an example what we've, for example, looked at. As you can see, that every symbol is one of our patients, and they scored themselves in how high they find their fatigue scores. And what's obviously really nice to see is that post-exertional exhaustion is really something that patients score really high. And if we compare the fatigue syndrome, the fatigue scores also with disease duration, we can see that that it's really well correlated with disease duration. So the ones who've got a high disease duration of ME/CFS also shown to have high fatigue scores. So a quick overview. I don't want to take a lot of your time, but I just want to quickly explain the conclusions we had of our publication, which we published early this year in Clinical Experimental Immunology, looking at comparing B-cell phenotypes of ME/CFS patients with healthy controls, and also the follow-up study, because what we wanted to do is to see if we see these patients for a second visit, is the B-cell phenotype still the same? And as Joe also addressed, we've basically set up an in vitro model looking at B-cell functioning, so the maturation, mitochondrial mass, proliferation, and differentiation. So you've seen this scheme before, but just want to quickly show that the first things that we did is phenotypes, looking at different subsets in B cells, where we found a significant difference in this early transition of B cells. And also, we also found difference in memory cells. First things that we found is that we found an increase in this marker called CD24 on total B cells, not only in frequency but also in expression. And this was mainly in the earliest stages of B cells, so the transitional ones, the ones that come early out of the bone marrow, which was really interesting. But the most important thing is that we need to see what does it mean to us, what's the functionality of this increase in CD24. So what we did is we cultured these cells and give them stimuli to see what happens, because we know that CD24 is basically really important for differentiation and maturation of B cells. And interestingly, what we expected as well is that if we don't stimulate them, nothing really happens, but if we give them a stimuli where the B cells proliferate and differentiate, we see that this mark of CD24 decreases. And interestingly, in unstimulated ones, in culture, we also found that in ME/CFS patients, this was higher compared to healthy controls. This is a second visit study, so we're still collecting samples, and we want to increase our numbers to make this really solid. So looking at phenotype is really helpful, but it's not a vital thing, because it will be really nice to see like a phenotype, but we really want to know how do these B cells react in these cells. And therefore, we propose to make some functional studies to see how these B cells, for example, increase their energy levels in different B cell subsets, look how these B cells proliferate. We've got a really nice assay set up looking at differentiation of these cells by cleavage of CD23, and therefore we can measure soluble CD23. And also wanted to see any antibody productions and possibly also autoantibody productions. Just a quick slide on what we've been doing, some interesting stuff looking at the mitochondria of these immune cells, especially in the subsets. And we did so by staining mitochondria on live cells by using Mitotracker, which accumulation is dependent on intact cell membrane potential. And this is really cool because we were able to visualize Mitotracker, so the mitochondrial mass in B cell subsets with flow cytometry. And as you can see here, this is the intensity of this Mitotracker. And what we found is there was, in B cells, there was a certain population which got high mitochondrial mass. And interestingly, if we look at this B cell subset, we can easily basically put these in different characteristics and found that naive B cells have got lower mitochondrial mass compared to the memory B cells. And this is just in healthy controls, basically showing the energy requirement of memory cells compared to naive cells. So as a conclusion, basically with the study, we confirmed the dysregulation of CD24 in ME/CFS patients. We are also investigating T-cell interaction with B cells, how B cells, for example, home to lymphoid tissues where they meet the vital connection they get with other immune cells, especially T cells. And the most important thing that I want to address is that we've basically set up an in vitro study looking at different components of B cell maturation. As I say, differentiation, proliferation. So this is a really functional study to basically analyze how these B cells work upon stimuli and how this eventually will react on, for example, antibodies or autoantibody production. So we've got these things going. We also got a new student coming to help us, looking at a new stuff that we want to do on intracellular cytokines, which looks really promising. And yeah, this is basically what we're really trying to do to get a good overview of what happens with these B cells in ME/CFS patients. So thank you to all the patients. Some of them are here, already met them. Thank you very much for donating blood because that's really vital for our research. But I really particularly want to thank Richard and Pia and Invest in Me charity for giving us the possibility to perform this research. And thank you for your time.
So Joe, could you come up and take some questions, please? I might say that to Joe and Fain are in touch with the Norwegian scientists who initiated a lot of this. So if people want to ask about how it's going and how they interact, it's obviously very important as part of the onward going collaboration. And it's European. So, who's going to start? Yes, do you see Richard? There's one over here.
Thank you. I just wanted to try and understand, so that if you give, can't remember, Rituximab to somebody with CEMFS, I'll get it right eventually, then you have to give it once and then you wait for 6 months and then you give it again and then you give it again. Is that how it works? And are there side effects that go with it?
I'm not a clinician, so I work with clinicians, but it depends on the response of the patient and it depends on the disease. I mean, there are many different regimes depending on the disease, what you're trying to achieve. In ME/CFS, the Norwegians are giving Rituximab like every 3 months. So the B cells will be away for a long, long time. And they get maximum responses sort of even later really than the rheumatoids, as far as I can see. I think we have to wait till till the results of this trial they're doing at the moment come out to really have a good look at the data.
There are plans to increase the interaction with other groups with the drug. You know, over the next few years, there's talks going on about duplicating it in other places, getting an international.
It doesn't always work. It's complicated.
But it's a serious drug, and so it probably has serious side effects too. You don't want to give it.
Infection in autoimmunity, no, and in cancer, the incidence of infection are not huge. I mean, you do get low immunoglobulins. Again, tends to be a bit disease specific. Lupus patients don't get low immunoglobulins, for example.
There's one over here. Yes, please.
Hello. I'd like to know when the UK Rituximab trial might start. And
Not
can
my
I be on
area.
it?
That's an easy one, Joe.
Oh, it's very hard. I mean, it's not directly to do with me, obviously. It's very hard to get trials going. I don't know if Joe wants to say, but it's writing a protocol for it to get it through ethics, etc., is quite difficult, and you need very, very committed clinicians in charge of this. But, I mean, I think things are moving forward towards having a trial. It's just getting everything takes ages. And I think it will go forward at some stage. But
Joe Edwards, do you want to say anything about your experiences at all with rheumatoid? No?
We didn't do a trial. No, we didn't do a trial, really. The drug, so usually they're drug company organised and funded. I mean, they've got buckets of money. These things are expensive, and also expensive in time. And the drug companies, when they come in, you know, they have people that do all these protocols, etc. And so really, it's quite hard doing any kind of trial without a drug company backing.
Is there anybody here from a drug company? Because when you do this at a cancer conference, 3/4 of the audience are drug companies, you know, all hanging about picking up ideas. It's very different, you know. But it can change very quickly, you know. People can suddenly get interested in something. And the audience next year may be half full of drug companies. We'll charge them double, you know. and no advertising rights either.
The thing is, also, there are other anti-B cell drugs too. That when when we've got more knowledge about what's going on, then we can design things much better to affect the disease. At the moment, it's a bit hit and miss.
One other question we've just time for. Yes, Richard, and then there's one down here, yeah.
I'm just wondering, it's very interesting to find out the role of Rituximab and the sort of research that's going on, but I wonder if there's any way in which legally patients can get access to Rituximab. I suspect I know the answer. But any legal way that their doctor can achieve this for them. And related to that, I wonder about how the severely affected patients can become involved in the research with Rituximab when they can't leave their houses or their beds.
Well, the answer to the first question I can't give really, because not being a clinician, I don't know what would be involved, but I'm sure some private. But again, it's difficult. You've got to monitor the patients, and then you need the backup of whether the patient's depleted properly, basic things. So really, it's not my area. But the second one, I know that the UEA are getting samples, blood, stools, etc., from patients who are housebound. So these patients are being investigated here in the Norfolk region.
Joe?
If I could just make a comment, there's no legal problem about treating somebody with rituximab. It's perfectly legal. The reason not to is that we don't know enough about the implications, and Dr. Fluger and Dr. Meller in Norway have quite specifically said that they do not want to encourage people to use it outside trials. They take responsibility for what's happening to people who are being given rituximab, and treating people outside that in uncontrolled conditions, I think their feeling is that that's going beyond what is a reasonable risk.
One last one. Is there anybody else? Sorry? Malcolm wants one. And then we'll have to move on.
Thanks very much for that. It was very interesting. But about almost a third of patients don't respond to rituximab. What are your thoughts on that?
Ooh. Well, there's not an antibody involved. I mean, it's the heterogeneity of the disease is a big problem when you're, looking at B cell function anyway. And I mean, it may be like in some lupus patients, you've got plasma cells that are not being depleted. Even if you assume that an autoantibody is pathogenic in all patients, which is probably the case in, say, seropositive rheumatoid, not all the patients will respond. We don't really fully understand that. It could be we're just knocking out the right B cells, or we're not getting rid of plasma cells either. So it's complicated. So even in a simple disease like rheumatoid arthritis, you don't get all the patients.
Last one, yes.
Just to support you, Henrik here. Just support the non-responder type in rheumatoid arthritis, 30% or 40%, and it depending on the criteria we want to use. Again, I think it's very worth telling people here, this is still on a project plan. Don't treat patients outside project at the moment, because we have to learn from this. Very important.
Yeah.
Okay, thank you very much. And thank you, Joe, and thank you, Fane.